All right, so just to recap what we have done in the past, we started. Hope I'm audible, right? Can you hear me?
Yes,
Yes,
sir.
sir.
Oh, thank you.
We can hear you.
Thank you. So we started precisely on the 13th of last month. We looked at phytoinformatics. So we have been able to pick a plant. We picked a plant, and what we did was to begin to mine the natural product in that plant. We made use of literature mining. Then we combined with database. So with respect to a given plant part, so with respect to the given plant part, either the leaf or the stem. So what we did was we created those datas. We took the names to PubChem, downloaded the structure. So we combined the, the the structures we got in several literatures. We had a benchmark of at least 50 literatures that has information on the bioactive compounds of our choosing plants. Then we mined about 5 different literature database, depending if the plant has been created in that database. We combined both the chemicals in those database and the ones from our literatures. Then we went to PubChem and downloaded all their structural data files. Probably there could be repetition, so we removed repetition. We cleaned the data because you will know when you sort, you will see the figures repeating after you have done the download. By the time you arrange them in one folder and then sort, you will see maybe 991 will repeat again, 991. Then you clean by removing the repeated structure. Our aim was to achieve up to 1,000 natural products from our plants, which we agree that it is achievable. Then when we are done with that phytoinformatics, we took a step further to toxicological screening. First, we have to convert those compounds using open babel, converted them to their SMILE code. We got their SMILE code, and then we use open babel to convert those structures to their SMILE code. Then we now did screening for their toxicity. We had about 17 toxic parameters. So we screened those are compounds. We used ADMET Lab 3.0 because it was more elaborate. That's about 17 major toxic parameters. We built a model, a simple model with our SF function to screen those compounds according to their toxicity. And the result shows that just a few, based on our criteria, we are not, in fact, we just shows that all the compounds we had, we are toxic based on our criteria. Then we are a bit, we did the percentage and we choose 70%. Then we saw that just a few passed that 70% test. That gave us idea that majority of our plants is toxic from our training. So it is a core that for us to be careful while using it in our formulation. After that, we went further for drug likeness screening, lead screening. We said, let's check if there could be potential lead, potential drug-like natural product from this plant. We did that. We subjected all the compounds into the ADMET screening also. We picked the drug likeness properties. We combined all the properties found in Lipinski rule, Weber, Gauss, Egan, and Muger. We combined all their properties, developed our own model. I believe the model we used in our training was Hendeb model. We did that, and we had some drug-like compounds. Then we went further for bioinformatics, which is where we decided to work on a disease. And we said you must study the pathogenesis of that disease, especially at the pathway level, cellular level, and the molecular level. So I gave us 3 basic key areas that we must focus on while studying your disease. I mentioned the biological process, which talks about the pathways of that disease. I mentioned the cellular component, which talks about where in the body is that disease targeted. Then I mentioned the molecular function, which now explains the function of those enzymes, genes, their role in the pathway, the role of the genes or enzymes that are involved in that disease, their role in the pathway. Let's know if we focus on those genes. We need to know them. A better explanation to this can be if we're able to do a Keck pathway. Yes, Keck pathway will give us a broader explanation to that. But we said that is done in a different training called network pharmacology. We explain how to get our protein, and last session we did bioinformatics, got our protein, prepared the protein, downloaded some of our bioactive compounds. So today we shall finish downloading those bioactive compounds and now do the docking proper, and after which we'll now look at the pharmacokinetics. Just to wrap it up, all the process we have taken before docking, they are all called pre-docking analysis. Then we now have the docking, and after the docking, everything you do after docking is called post-docking analysis. You can still call it pre-docking screening, then docking and post-docking analysis. So tonight we shall finish up where we. I'm going to share my screen right away. Hope we are together.
Yes, sir. We talk together, sir.
Yes. Yeah. So this, can you see my screen?
Yes, sir.
Good. So I downloaded about how many? No. This is not the document we are using. Drug light. Okay. Yeah, this is it. Or am I confused? Okay. So we are using the drug light. Remember, we are working with the 70% as our criteria. So let me just color it so we know what we're working with. So we are working with the 70% as our criteria. So we say let us download. We stopped at downloading the structures, our ligand. But I know we have downloaded some of them, about 3. We downloaded 3, which are these 3. The first 3 that we downloaded, and we later converted those. I showed you how to convert 2D structures into 3D, which is what we have on that converted 3D. So let me quickly now finish up the other download, or better still, we can work with what we have here already. What do you think? We downloaded this first three. Should we just continue with what we have? What we have downloaded, or we finish all the download?
Let's work with this, please.
Okay, we should just work with what we have now, because
Yeah.
even if you want to finish all the download, it's the same thing we are doing. Yeah, it's the same thing we are doing. Then if you happen to have a 2D download, it's the same thing too we did. You say convert 2D to 3D. So it's a training, is exposure on how to do it. Even if we say let us download all of them to see how the result will be. Remember that even the result we have here, this data was not complete. We had about 1,400 something data. We just pick 20 for the sake of the practice. So there is nothing else again that you can do which we have not explained. Just continue downloading all the ones that we are drawn like. Then if you have 2D, you can now convert the 2D to 3D. Now the next step is, because I'm using Pires. In Pires, you cannot highlight all your ligands at once. You must copy them one by one. If they are separated, if they were separate, like I cannot highlight all this once in Pires and then dock, then open. It does not permit that feature. It allows you to be copying the files, copy one file, one by one, open it one by one. So because of that, I'm going to compress all my ligands into a single file. So even if I have 50 ligands, if you don't convert them, let me quickly show you what I mean. Assuming you want to open a ligand, I will still come to this. If I come to this place, all right, I go to my download where the files are. Now you see that I cannot highlight all at a time. I can't highlight. I'm trying to highlight all my ligand, but it's not working. That means I have to copy one, open. After opening it again, I go again, copy another one, click another one, open. So imagine if you are docking 50 compounds at the same time. Do you have that time to be doing this for 50 compounds at the same time, or 100 compounds at the same time? So the best thing you do now is to compress all those your 100 ligands you want to dock, or 50, into a single file. Just like what we observed when we converted our 2D structures to 3D, it kept those three structures in one file, which is this converted 3D structures. So what we'll do now is to use our Open Babel. So can you launch Open Babel for me? Can you launch Open Babel? We use our Open Babel to compress our ligands we want to use for docking into one single file. That is what we do now. Into one single file. So we'll come to, now you make sure the input is in the right file format you want to upload, and you also make sure that the output is in your right format, which is SDF. So the next thing you will do now is you come to this your input place, your input format, click on these three dots. Then Open Babel can allow you to copy all those ones you want to convert. So I will take my converted 3D. All these ones, all these 2D has already been converted into this converted 3D for my last station. Then I will now highlight the remaining ligands, which are my negative control, my 2 positive control, and my native ligand. So if I open them, the idea is I want to compress them, I want to cluster them into single file. They come to the outputs and save it. Choose the name you want it to be. I can say clustered. Can say clustered ligand, clustered ligands for docking. So whichever you want to put it. Once you click on save, don't choose anything under this convert. Don't choose anything. Just go straight and click convert. Just go straight and click on convert. What that does is that it just compressed. There was no conversion that was done because we did not choose any conversion pipeline to be done. So we just click on convert. So it will convert those single files into one file. Those separate files has been converted into one file. And that one file is called the cluster ligands for docking. So you can now go back to where you saved that. That is on my download. So this is it. Clustered ligands for docking. So there are 7 here. If you open it in your Discovery Studio, you will see those 7 clustered compounds from your Discovery Studio. So we have succeeded. We have our ligand prepared. We have our protein prepared. So the next is to go to docking. We'll now go to the docking. So I have my Pyrus launched. Can we launch our Pyrus? So can you launch your Pyrus? Now, why I prefer Pyrus to some of these softwares like they commonly use Autodoc. Pyrus it has 3-in-1 software. It has Autodoc Vina, and OpenBabel. And then Pyrus can allow multiple ligand interaction with a single protein, whereas the conventional Autodoc only allow single ligand for docking. So when that process is done, you go again for another ligand. Now, Pyrus can accommodate as much as you can. But please, we will not say because a lecturer is good at what he's doing, you now decide to over-labor him by giving him much courses. You will see that large courses will affect his performance. That means Pyrus can dock 100 ligands at a time, but don't give him 100 ligands. Please, if your mic is on, kindly mute your mic. You will not say because your phone is a 150 gig. Then you want to allow the phone to be filled up to 150 gig. You now notice that the performance of that phone will begin to reduce. So this same thing is applicable to Pyrus. At least I advise you to work with 50 ligands per time. So if you have multiple ligands, cluster them separately. You can cluster first 50, cluster another 50. So that will be it will be faster. The docking will be very fast. But if you take all your 100 compounds, 150 inside Pyrus, it will be so slow because of overload. Now, the first thing we'll do now in Pyrus is to load our macromolecule. So the protein is your macromolecule. We'll import our protein into Pyrus. You go to File. This is my cursor. Go to File. Are we together? I would like to be faster than others. Are we together?
Yes, sir.
All right. So you go to file. Once you are at file, you click on file. You will find load molecule. You're going to find load molecule. So click on load molecule. What we are doing is to load our macromolecule, our protein. Then locate the folder, the file where you kept your macromolecule. So this is the one we saved. The 1086 hypertension. You click on it. Then you click open. So it has opened. You can see the 1086. But then this protein is not yet in the pyrase. Remember, this protein was in PDB. So we are going to convert it to PDB-QT. That is the file format that pyrase understands. So because of the open babel, open babel can do file conversion. So the open babel in pyrase will now enable the file conversion from PDB to PDB-QT. So click on this protein, right click. Once you right click, you come to Autodoc. You will see make macromolecule. So pyrase is very simple and it does not need programming, does not need writing of program. So it's a simple thing to use. So click on make macromolecule. At that point, is making Autodoc macromolecule, meaning that it's converting the protein from PDB to PDB-QT. And once that is done, you are going to confirm if it has converted. Quickly look above, you will see Autodoc. We want to see if it has been converted to an Autodoc molecule. Once you click on Autodoc, you will find the protein here under macromolecule, which simply means that that protein has been converted. Then the ligand is still empty. If you click on this small intersection here, you will now see that the protein is in PDB-QT, which implies it has been converted from PDB to PDB-QT. All right. The next job you are going to do is to load your ligand. But please, from experience, after you have import your protein, kindly close your pyrase. I don't know, but most of the pyrase and even in diverse systems I'll be using, if you have been working frequently with pyrase, there will be a time when after you load your protein, you try to bring in your ligand, it will cut off. So to avoid that, once you load your protein, then close the pyrase, then we open it. So even if you reopen it, what you have, the protein you have imported will still be there. Yes, the protein you have imported will still be there. Pyrase is an open source software, so you don't blame it. So let's just give it a little time while it opens. All right, so I was showing you about those ligands that we clustered. So these are the ligands we clustered. The 7 clustered ligands. All right, so I close it. The pyrase takes little time. It all depends on your system. Okay, it has opened. So if you go to Autodoc, you will find that the protein is still there. So at this point, you are going to import your ligands. If your mic is on, kindly mute your mic. You are going to import your ligands. Just come a bit down. Look at my cursor. Come down to where you see open barbell. Click on that open barbell. Then move your cursor towards the right end. You will see the first icon with that blue, with the green plus sign. The first icon with the green plus sign. Sorry, I need to know, are we doing this together? Or we are waiting to get the video, then later we practice? Hello?
Yes, sir. We'll practice this because we are lost.
That was why I asked, are we together? And
practice.
the reason I got was yes. So even those who were lost also said yes. I'm even very slow. That is why I was asking. I always ask, are we together? Some persons will say yes. Why those who are not together will keep mute? We have also said no. Then I will know where you are and I will know what to do. But well, the video will be available also as normal, so you are going to still take it up from there. Okay? All right, thank you. So we come to, when you have clicked on open barbell, you come to just take your cursor towards the right end, the first icon here with green plus sign. That is telling us to insert new item. So we shall use that function to insert our ligand. So I click on insert new item. Go to download. That is my cluster ligands were in the download. I can find cluster ligands for Docker. I open it. All of them will open, those 7 ligands. So the next thing you are going to do now is you are going to minimize the energy. The essence is a molecule with a higher energy takes longer time to react. But if we reduce the energy, then it can react faster. So, and this energy are always reduced. In fact, energy minimization is usually done with the help of what we call force feeds. And Pyres has a force feed called UFF, which is universal force feed. Those that were with us during the beginner's course, we minimize with ChemDraw, but ChemDraw was using a MM2, which is molecular mechanism 2. But Pyres uses UFF. So we don't know reduce the energy, so the reaction can be faster. Because if the energy is high, that means that reaction system must wait until it generates that energy before it will react. So you can imagine a reaction that requires 2 joules and the one that requires 50 joules. Which one will be generated easily? The 2 joules can easily generate the energy and react. Whereas the one that has 50 joules will still be waiting. Maybe at the end, it will end up begging for energy from the surrounding. That is endothermic, begin to beg for energy. So it may be possible that it may not even acquire the energy needed. That kind of reaction is not feasible. So we shall now minimize the energy by just click on any of the structure, anyone. Once you click on any of the structure, you right click. When you right click, you will see minimize all. So you click on minimize all. It will minimize all the energy. And as that minimization is done, you'll be seeing the energy of minimization attached to the CIDs. So here you are seeing UFFE, which is Universal Phosphide Energy 344.65, which is usually in change in G, in delta G. That is this free energy. Those with little chemistry background from secondary school, let me not secondary school because we must have forgotten everything we did in secondary school. From Team 101, we must have done stuff like free this energy, which is the energy available for work. Now, the next thing you will do, remember that these ligands were downloaded as SDF. Iris has autodoc, and that same autodoc that we used for the macromolecule does not understand SDF. So it will convert that SDF to its own language, to PDBQT. So also right click on any of the ligands. You right click on any of the ligands. You see convert all to autodoc ligand, PDBQT. So you click on convert all to autodoc ligands, PDBQT. Everything will be converted to PDBQT. So, and once it's done, you will see a table shown above here. This table is both the work you have done before and the current one you are doing now. So I want to see a table shown up here. This my top right hand. That shows that it has been converted. But if you look up here, ligand is still showing nothing. You will see them appearing under ligand. The next thing you will do is to come down beside the open barbell, the first one. You will see Vina Wizard. Click on Vina Wizard. Click on the Vina Wizard. So once you click on Vina Wizard, you see start here, select molecule, run Vina, and analyze result. But the first icon, which will be empty, showing is start here. So we are going to start here. So what you will do is click on start, come down to the bottom right of your screen, bottom right, the right hand, the ending, the bottom right. You will see start. Click on that start. So click on the start. Once you click on the start, you will notice that those your ligand just appeared. They just appeared below the ligands. So we have our ligands in PDBQT. We have our protein in PDBQT. The next thing is to dock the ligands against the protein.
Excuse me, sir.
All
Excuse
right.
me, sir.
Yeah.
Yeah, good evening, sir. So,
Yeah. Good evening.
my name is not, the names of my compound are not appearing under the name. Here where you have the
The names
name
what? What
sign
are the names?
created, it's just like underscore UFF, then the
The one is
number.
not showing the CID number.
No, it's not showing the name.
No, it's not. There's no name. It's showing number like 53C7 something.
No, I changed my CID number to the name of the compound.
You change your CID number to the name of the compound.
Yes.
Okay. Well, for this training, we didn't say so, but I would like to know, how did you change it?
I when I downloaded it, I just clicked on it and pasted the name.
Okay. Can you see my screen?
Yes, I'm seeing the names of your compounds also. Some are names, some as the CID numbers.
No, don't bother what you are seeing on the table at the right. Let me see. Let me understand what you say you did. Can you see my cursor?
Yes.
You mean
So
you came to this place and you
click
renamed,
on it,
right?
click on it and then paste just Control V because I already copied the name
So
from.
you change the name to whatever it is, like uh,
Yes.
acetosine benzene, something like that,
Yeah.
right?
Yes.
Okay, I'm coming. Maybe let me just state one name. And what was the next thing you did now?
That was it.
Okay, there you before you
Enter.
came to Pirates now,
Yes.
and you are not seeing the CID number. Can you see this? Okay, when you opened it, when you open the light down, before you minimize, where the number show you.
When I opened the ligand,
Are you joining with your laptop?
yes. No, I didn't join
Share
my
your
laptop.
eh?
I'm joining with my phone, but I can take the picture and send it to the group.
Okay, you can do that. So while she's doing that, the next thing we shall do is to select all our ligands and the protein, then give a command to start docking. We'll now give a command to start docking. So for us to achieve that, okay, sorry. So for us to achieve that, you hold your control key in your keyboard. Hold your control key on your keyboard. So your left hand, just hold the key, control. Don't move your hand from there. And then okay, Adolphe, I be so you have seen what you sent. I will attend to that. Then click on the first lie down, click on the second lie down, click on the third lie down, then continue clicking down. So as you are clicking down, it will be opening down. Now, as you are clicking, if you look at this white space, see my cursor, you will see 7 lie downs selected. That means I've selected the 7 lie downs. Remove your hand from control. Then click. Now, if you watch, you say no macromolecule selected because I've not selected any macromolecule. Then click on the protein. Once you click on the protein now, it will show. Now, sometimes if you click on the protein, it may not show that you selected it. What you will do is you come back to this intersection here, just beside the protein, right click there, then the protein will show with the PDB-QT. Then you click on that one. So whichever way, when you first click on the title and it did not show, then click on this intersection, you will see this the file, the PDB-QT file, then click on it, it will come up. So we have selected 7 ligands and our macromolecule. The next thing that you will do is come down to the bottom right, and click forward. Once you click on forward, you have sent the word for docking, but docking has not started. Now, one thing that Pirates does is that, let me show you.
Sorry, before you click on forward, what did you do?
No, once I click on forward, it will now appear this. This will be the next thing
No,
that will
before
appear.
you clicked on forward, what did you do?
Okay, I first click on start. Are you with me?
Yes.
So when I click on start, I held my control key in the keyboard and selected all my ligands and also selected the protein. Then I now click on forward. Did you get that?
Yes, sir. Let me do it.
All right, thank you. Now, if you look at this,
Oh,
my
no, sir. No, sir.
eh?
Sir, please come again. Sir, please come
See what
again.
happened now? Look. Can you see my screen?
Okay. Yeah.
All right. I click on start.
Okay.
Once you click on start, you then hold your control key on your keyboard.
Control key. Okay.
Then start clicking on these your ligand, all these ligands. Start selecting by
Okay.
one. It will show you blue. Be selecting them one by one until you select all of them. Make sure your hand is on the control key.
Okay.
You will see here 7 ligands selected. I have selected my 7 ligands. Now, even if you have many ligands here, I can have 10 ligands, but it's the ones I select that will dock. The ones I just select dock. Yes.
Okay.
Yeah. So after I selected it, now come to your protein. Under it, you can find macromolecule. Click on that macromolecule.
Yeah.
Once you click on the protein there, the 1086. Once you click on the 1086, the next thing you will now do is that same bottom right where you click on select, you now see forward. You see forward.
Okay.
So click on the forward, and that is where we are.
Okay. Thank you, sir.
You're welcome. I want to explain something. Pyrase automatically identifies the binding position, except you want to change it. Can you see this box? Oh, you can see this box here. Now, the box here is called grid box. It's called grid box. Grid. GRID. That grid box, that space where the grid box covers is the only space where the docking interaction will take place. So this grid box, so that means all these amino acid structures outside the grid box, the docking will not occur at that point. Those parts will not be among the docking. So this part defined is what you use for the docking. Now, if you want to change the grid box, then you can change the values here. You can reset. You can reset. You click on reset. You can reset the values here if you want. But this is automatically a defined binding site. Then, please, you have to copy out this center value, S, value of Y, and value of Z. Because while you are doing your docking, you must specify the grid box dimensions and center value. That will be in your methodology for docking. If I see a paper that is talking about docking and there was no grid box dimension and center value, like no grid box dimension, I usually throw away that paper because you did not specify the region of your interaction. The essence is for validation, for redocking. So if someone wants to validate that result, want to redock, it means that if I want to redo what you have done and I want to expect the same similar result, there is no high. See, if you dock to the same molecule on a protein, dock this one, come again and dock again, do you expect to have the same value, the same result? Do you expect the same result?
No.
No, no.
Yes. We don't expect the same result. That is true. So don't get biased or confused now. But the difference will not be much because these docking results are not exact. They are prediction. Everything about in silico is all about prediction. It's prediction. But with high precision. With high precision. So if you define the grid bus, it means that if I want to validate your result, I will do what you have done with your grid bus. Then I should expect something similar. For instance, if your binding score was 6.2, I can have 5.9, I can have 6.1, 6.3. They are all close, they are all within. So it means you are right. That is why you must specify that grid bus dimension for validation, for reducing purpose. So you must copy these values out, copy them out on a paper and keep, because you are going to record it in your methodology. The next thing you will do is to click on forward again. There's another forward still showing there. Click on forward again. At this point in time, you have sent a command that let each of the ligand begin to dock with the protein.
Question.
Yeah, question.
Okay. We are talking about
Yes, sir.
this grid box number. So assuming I want to validate now, where will I input? And I'm reading a journal. So and I saw this grid box dimension. Where will I input it? And I want my doc to happen exactly
Okay, please,
the same.
I didn't get it. Can you be a bit audible?
Hello, can you hear me? Assuming
It's better.
I'm reading the paper. So, and I want to validate the grid box dimension using Pyrex. Where will I input this number?
To input which number? The one you saw somewhere.
Yes.
That is the grid
I
box
want to
dimension.
release it. Yes.
You reset. So maybe you saw a work. You want to validate that work.
Yes.
So you saw a work. Maybe they docked acetobenzamide with 1086, and in that paper you saw the binding affinity. So you want to redock. Then you prepare the protein the same way using the tools that they use. Because while redocking, I will encourage you to use the same tools that was reported in that work, number one. Yeah, because these tools differs. That is why your report must bear the computational tools you use in your work. The results you have when you use Pyrex may not be, you don't expect the same value when you use Scrundiga Maestro or when you use Autodot Vina, Gold, Glide, and others. But they correlate the same thing. They all correlate the same thing. So, but if you want to use Pyrex now, and those people specified their grid bus, you come to this place now and do reset. Once you click on reset, with the help of reset and maximize, you can then adjust the grid bus dimension to the one you saw in that literature before you proceed for the docking. That's what you will do.
Okay.
Because if you continue with the one showing here, it will not be the same. So with the help of your reset and maximize, you can adjust these values. We adjust these values, then you now set it at the S value, Y value, and the Z value that we have reported. You proceed with the docket. But make sure, I do recommend you validate with the software that was used in that study. Like for instance, I cannot do a work with GCMS. I want to validate that same result. Now you're using HPLC. So that is not the whole analogy. Now, you will see that, yes, is there a question?
Yes, that's great, boss. Why didn't you adjust or expand it? I was thinking you would probably do something to it.
Yeah, I did not do anything with it because Pyrus automatically understands the active binding site of your protein. See, if you load 3 proteins into this Pyrus will not give you the same grid bus dimension. It will give you different grid bus dimension, and it understands is being programmed to understand the binding site of that protein. So that is automatically generated by Pyrus. So there will be no need for me to adjust, except I have a binding site. Except I have a grid bus I want to use, then I will adjust it. Now, so you can use yes, that is automatic. Yes.
Okay, sir.
Now, you can actually use Discovery Studio to also find out the binding site of a protein before you proceed. And oftentimes, when I use Discovery Studio to check for the binding site, the values usually correlate with the one in Pyrus. The difference will just be maybe the Discovery Studio might give me 23.46. Pyrus will not be 23.45 or 23.5. So the difference is negligible. It's not really much, because you can still use Pyrus to get the attribute. You can use a Discovery Studio to get the attributes of your protein, because it's attributes. Maybe when we shall organize a specialized training, this is centered on macrognosis. So we bring out a part from each of these areas that is essential for macrognosis and give to you. So we will not start teaching each of them as a whole, as a course. We'll get the parts you need in macrognosis and give to you. So that is why. If not, there are a lot to do with Discovery Studio. DS is not only for file conversion, for visualization. No, I use Discovery Studio also for macroform modeling. If you look at the feature, you will see a lot of things it has, but we will not start bringing all those ones because it does not correlate with what we are doing. You understand, right? So we have to just get the part that is needed for what we are training and review that to you. All right, thank you. Now, this docking, you see, is reading. Once one finished, then the other. If you have a good system, it will not take you longer time and you are done with that. Or if your system is not overloaded, so what usually affect this is loads, your RAM, and then your processor. So a good processor will we run it as fast as possible. Now, this docking is running 9 times.
One hand is up, sir.
You say?
Dr. Fisher want to clarify something.
Okay, oh, sorry, I'm not seeing the interface. I'm just on my screen. That is why. Sorry. Okay, sir, please go ahead, Doc.
Okay, thank you very much. In
Yeah,
relation to
Doc, so sorry. I'm not at the meeting interface. I'm on my screen. That is why.
okay, sorry. I want to just ask a question in relation to the last question. Now, this docking that you have shown to us, and then of course the position of the grid bus, I wanted to ask, how does that relate to blind docking and site-directed docking?
Okay. All right. In blind docking, the active site is not defined. The active site is never defined. So what you will do is you can now define an active site by yourself. You can adjust because you want to use that approach of blind docking. So since you want to use the approach of blind docking, even if Pirates now gives you an active site, though I've not bothered using blind docking, I use it often when I just want to do trial by error, like I just want to try something myself. So you have to now start using different active sites. You define one active site and you dock. You define one active site and you dock. But Pirates cannot automatically allow you to set your dimensions at 0. But MOE, which is molecular operating environments, MOE has that feature of blind docking, where the dimensions will not be specified. It's a feature. It will not be specified. So MOE understands that if you choose blind docking approach, then there is no XYZ value. It is now after the docking that a particular dimension will be provided, because at that point the ligand was able to interact at multiple positions to achieve a better site. So it is now that site that was disabled compared to others that you have that the grid bus dimension will be provided. So, but if you're using Pirates now, you must define the binding site. So it does not give room for that blind docking where you don't know the binding site. So even if you don't know the binding site, you must set a dimension. You must set a dimension. That is Pirates. But if you really want to do effective blind docking, then go for MOE. MOE is one of the best tools when it comes to docking. It's far better than Strundida. It's one of the best tools. The thing is that it's expensive, very, very, very expensive. The last time we used MOE was as a team, and we got the license. That was about $3,000. So you should know what $3,000 means in Nigeria. And you want to do a work as an individual, you are buying a software, one software for $3,000. And it's not even a lifetime, you renew it yearly. So what we did then was group of researchers who have interest. They contributed and we got that license. And after that year, we are unable to renew it, and that is how I stopped using MOE. But MOE is the best. I love using it for blind docking. It will be very good for blind docking. And since then, I don't do blind docking again. Thank you, Doc.
That's okay. Thank you very much.
Yeah. But we can still get some of these licensed softwares through our grants. Through our grants. Because through grants, is part of your the tools you are using. So you can get a grant that will give you that opportunity of getting some of this licensed software. Or teams or government. If you know someone in the government, they can sponsor you. But imagine giving you $3 for just one software. All right, thank you.
I have a question.
Yeah, go ahead, sir.
All right, thank you, sir, Doctor. I have a question. For example,
Yeah.
now, if I'm doing a docking, like 5 different extracts, like NXN extract, water extract, like that, up to 5, then focusing on a single target, a single receptor. So why is it that for ethanol extracts, after the docking of the phytoconstituents from that particular extract against that protein, then my reference drug, for example, is having the maybe minus 7.50. Then the same standard and the same protein from other extracts, like maybe propanol extract, for example, now, is
Yeah.
now having different value compared to the first extract. Still the same standard and the same protein.
Yeah. Yeah. I think I mentioned that case a little, but not directly. But then, have you observed that?
Yes, yes.
Okay. Now, if you observe
So,
that,
recently,
can you tell
last
the
week.
vibration in that control over the extracts in each of the extracts, the vibrations, were they much or very close?
They are very close. For example, there was an extract. I use a probably coconut water extract. It was
Yeah.
given to me because I'm just helping them to run it. They are paying me some tokens, right? So that coconut water extract, there was a compound there that had minus 7.10. Then the reference drug minus 6.90.
Yeah.
Then other compound minus 5 point something. Then in another extract, water
The
extract,
control, the
water
reference
extract,
dot had what?
add a minus 7.2 something,
Okay. No
followed by
problem.
minus
Uh,
minus 6.7, 6 point something.
yes. And the issue now is why will the same reference dot for the same protein be giving different values, right?
Exactly. Yes, yes.
Yeah. Now we said it here. Yes, I asked that question that if I do a docking, that was when I was explaining about docking. And if I redock again, I asked, do we expect the same binding affinity? Somebody answered no, which is right. Then I justified the answer by saying that computational studies is based on prediction. Or any result generated from computational study is based on prediction with high precision. It's based on prediction with high precision. And the percentage error is even more than the normal 5% we allow for lab work. You know, if you are doing a lab work, I assume, in fact, we accept a result of 95% confidence limits.
0.05.
0.05.
Yes, yes.
Good. But in the case of a silico, the acceptability is not up to that 95% though. I can give it 80% because it's prediction. That is why do it over again, you won't expect the same results. It's working by algorithm.
Okay.
It's working by algorithm. It's working with equation. Some of these things were built with Schrödinger equations, wave mechanism models, contour mechanism principles. So that was what they use in building this thing. This is an instruction. That is the problem why some professors will tell you, I don't accept all this on a computer result. You don't go
They will say it's not. They will say it's not reproducible.
they will say you don't carry computer, but they don't understand the algorithm. They don't understand the process behind it. Now, it predicts according to the activity. So if you have a higher prediction, it's suggesting. Let me say it again, that any result you get from silico is a suggestion. It's suggesting that for this compound A to have this kind of result compared to the control, I'm thinking that it will be very, very better. It will be a better option. So what do you do? It gives you an idea. So you can now go back to the lab and validate that suggestion. It's just like hypothesis. You develop an hypothesis, giving a guess, a reason to an observation, but you have to validate that your hypothesis by conducting an experiment. That will be design an experiment according to the hypothesis. So if the experiment gives you positive, you accept the hypothesis. If it does not agree, you go back and reject and develop another hypothesis. So it's suggesting. That is why we preach every time that please validate. We had a training on green chemistry. We had a defense. One of my mentor, Professor I.E. Otokere from Michael Okpara University of Agriculture, Abia State. All the work that we are presented, he made same comment, which is go for validation. Validate your work. Validate your work. Some journals will not accept it unless it is validated. But see how good it is. If you discovered 100 compounds in a plant, and you want to identify which of these 100 compounds has the potential of inhibiting a target, will you now start isolating each of those 100 and begin to test their inhibition? But when you now subject this 100 into in silico studies, it will now suggest a few from the 100 for you now to focus on that few and go back to the lab. That is why we also say that a compound might have a higher binding result with a protein. That doesn't mean that that scenario is what happens inside the body. Because by in silico, that compound binds with the protein. But if you eventually take in that compound, it may not even have business with the protein. That is the essence why they now introduce network pharmacology to handle that case. So we can identify a compound and the exact protein that it can inhibit. If I have 20 proteins, my compound can bind with all those 20 proteins. But in reality, that compound cannot be effective on those 20 proteins. The compound might even give you high results for all the 20 proteins. But in reality, that compound might not even interact with all these 20 proteins. But Dawkins says that it has better interaction with these 20 proteins. So because some of the proteins are off-target interaction, which is not necessary. So the compound may not interact with those proteins. And again, after your docking results, you have to check the stability. If those compounds were actually stable, at that receptor site, which is why we do MD. Can it last? How long can it stay? So, because if you take that compound to the human body, will it just meet the protein and leave the protein, or can it form a complex for long?
The
So
visitor.
that is why you now bring in MD. So if you see that the MD result shows that compound, the stability is poor, the
What's the
flexibility is
meaning of
poor,
empty?
even if you have -20 interaction, it simply just shows that there is poor stability. You know what we do to do in thermodynamic stability. For those who have idea on physical chemistry, that I've done a lot of lab work on thermodynamic stability. So such kind of experiment is what we are bringing with MD. So that was the reason why MD came in, for thermodynamic stability. MD is just based on thermodynamics, stability of the changes of that protein and the ligand. So if MD says that, see, this thing does not last long here, is giving us a suggestion that these bioactive compounds in this protein may not be. In fact, you may have a ligand that has a high binding result, but may not be stable with MD. Whereas the one that has a lower binding result becomes stable. So the stability in MD does not correlate with binding affinity. And that is the reason why, if I'm choosing my heat, if I'm doing a work and I'm choosing my heat ligand, I don't just use first two of. Some persons relies on the first one as their heat. Sometimes I pick first 10 or first 5 as my heat, because I don't concentrate on only the first one. So if I pick this first 5 as my heat, I will then subject this first 5 into pharmacokinetics screening. Then you may also notice that even that one that you are celebrating, when you do a doctor, then you see -12, -13. Wow, you shout. But when you now subject to kinetics to PK studies, that compound just failed the PK criteria. You throw it away, or you now go for lead optimization. Even when the compound also passes, you still have to do lead optimization. So docking result is prediction. If you do it over and over again, you will not have the same value. You may have similarities. Lastly, let me even tell you this now. I had a training July last year with students, final year students in Anambra State University, now Chukwemeka Odumegwu University, students in biochemistry, final year students. I was to handle them. They were 11 in number with a various system. We docked the same thing at the same time with pirates, the same thing, but our results were different. So the students now asked me, sir, hey, my result and your result and her result, they are not the same. I say, yes. You don't expect it to be the same, but if you look at them, the one that will give me highest will be the one that will give her highest. And the values will correlate. There will be resemblance in the value. The variation will not be much, because it's all about prediction. Thank you.
Thank you, sir, for the enlightenment. Thank you so much, sir.
Thank you. So don't put all your hope and mind, your soul and body. It's giving you idea. I want to do something. While we are waiting for the docking, we are supposed to end this class today. But this is already 9:21, and thank God the docking is almost getting. So can I give you extra 1 day? Or we should end everything
Yes.
today?
Don't end. Give us extra one day.
We hope for the best.
I want to learn more.
Let's see others reply.
Give us extra
want-
one day, oh, sir. We want to learn
we want-
more.
What do you want to learn more again?
Extra one day, sir. Extra one day.
Sir, like
Okay,
what
let's
you
have
just said now, I wanted to ask a question. Maybe when you are done.
Okay. Now, the reason is we still have to do post-docking analysis, like the visualization. I don't want to leave any, I don't want to take because of time, and then I leave some necessary things that we are supposed to cover. If possible, we even extend.
Please,
Right.
I'm hoping that you will take us that molecular dynamic, especially that homo thing. I really want to learn it.
No,
I
MD
really
is
want to
not
know about
here.
it.
Jesus.
Please, even though it's just introductory, please. That's
MD
the
is
reason
not
why I
here.
joined this class. I know all of this except for that homo.
Which one? Homology modeling.
Yes.
Yes, even
Please,
network morphology, homology modeling, they are not. We can't pack all of them to this training, but keep watching at our advert. We have a training this year also for homology modeling.
because I am working on a particular research now. The only thing that has made me not publish that paper is that particular thing.
Okay, don't worry. You publish it this year because I may not fast forward it because we have a calendar, but I know we have a training on homology modeling this year. Yeah, we have a training on that area is very unique. Very unique. I heard a report. I saw a report in LinkedIn that one of our Nigerian researchers uploaded a protein data bank. That was early this year. So that was really is a good innovation. Him and his co-researchers, they saw that protein was not is not in PDB. So that means they did all the genomic studies, proteomic studies, metabolomics, and all those stuffs. Got the amino acids sequence and so much. Did the homology modeling, crystallized the protein, and probably they wrote to PDB that they have the protein. And if you look at the authors, their name will be there. Let me even search for that protein. You can. Somebody showed. One of our participants here was the one that brought that to my knowledge. Mr. Imran Salusi. He brought that to my knowledge. So I can still search it.
While we are waiting for the docking to finish and while you are doing your search, I have one question. May I go ahead?
Yeah, go ahead, Prof.
Yeah. The docking process that we've been talking about, we've been focusing on endogenous macromolecule as a ligand. I know that one of the very popular area of research in our universities is metal toxicity. And the number of plant products are being investigated for their chelating property. Apart from metal toxicity, you can also have other exogenous compounds or molecules that get into the body that we want to bind them with. So my question is, for docking, if you want to use the docking strategy to develop drugs along this line, all these protocols that we've handled so far, is it the same thing when it concerns exogenous molecules? All those ones that we've used of endogenous macromolecules, or there is in the approach for docking against exogenous macromolecules, are there peculiarities in the docking process? I hope my question makes sense.
Okay. Yes, I tried to get that, but there's a condition for you to use docking, depending on the software you're using, though. like if you're using pyruvate now, that compound, the ligand must be a small molecule. It must be a small molecule. If you're using a large molecule like peptides, this doctrine does not, pyruvate does not with peptide, because peptides are protein. But MOE still does protein docking. What I'm doing here is ligand protein docking. We also have protein docking. So I don't know if that is the area the question is coming from.
The question is talking about not ligands, but as targets. Targets, for now, we are looking at targets, derived from endogenous molecules, receptors, enzyme, and so on and so forth. But what I'm talking about is in case that our target is not an endogenous molecule, but an exogenous one that is getting into the body. For example, metals, metal toxicity like lead, arsenic, and so on and so forth. In that case, our ligand is going to be binding with the metal for chelation to get it removed from the body. And the pattern of interaction is also about, lock and key model. That is binding. So I don't know if docking is also used, can also be used, in developing drugs for treatment of metal toxicity or any other toxicity that is caused by small molecules.
Okay. I got that based on that clear. I did a work. That work was published 1st of November. No, that work was published November 1st November last year. It was purely on metal toxicity. In that work, albino rats were intoxicated with mercury chloride. They induced mercury chloride in albino rats. And then after the induction, we assessed some organs, some glands. We assessed the neural glands, like the thalamus. Then when we assessed, we saw that there was an impact of that mercury. So we asked, okay, can we use docking to do a work that we assess if these bioactive compounds can reduce that metal toxicity? I believe this is the angle this person is coming from now.
Yes.
So we use aloe vera. So can we see if aloe vera extract can mitigate that mercury toxicity in the brain? So when that work came to my table, what I did was, there are some enzymes. I will give you two approaches to do that. There are some enzymes that binds with those metal. There are some enzymes that binds with those metal. They are called metalloproteins. These metalloproteins binds with this metal. And once they bind with this metal, they become toxic. So we are now assessing the potential of these bioactive compounds to bind to that same metalloprotein, such that if these bioactive compounds be able to have a higher binding results, then there is a tendency that they can knock off the metal attached to those protein. So they can knock off the metal attached to that protein. And if that is possible, it means they have the ability to cleave effectively on that metalloprotein, to cleave to that metalloprotein. So if they can do that, then those ligands can be used to mitigate that effect. So when we did that docking study, we saw that some of the ligands were able to bind with this specific metalloprotein. There are proteins that bind specific to mercury. There are proteins that bind specific to arsenic. So you look for those proteins. Then see if your ligand can bind effectively with those proteins. When the result shows good, we went back to the lab, took those extracts, and we inoculated, we injected those extracts to the same amino acid, the amino acid that has been induced with this metal. We noticed some change in the histology in those their histological studies, their histology graph or whatever they call it. So there was a change because we administered different dosage. We now notice reduction in the toxicity, which aligns with what we did in docking. Now, the other way you can do this now is you can now do ligand metal docking. But Pyrus does not do ligand metal docking. MOE2 can do ligand metal docking. That is to see the ability of my ligand to form complex with this metal. Then, if you can get access to this MOE, then it will be best for you to do ligand metal docking. Or you look for the enzyme, the protein that binds to that metal in the body. Because for the metal to become toxic, it must interact with an enzyme, with a protein, with a receptor in the body. You identify that receptor in the body and interact with that receptor. That is what we are doing now. And I have done a lot of work for people on that aspect. I've done a lot of, mostly for those in anatomy and physiology. They are the ones that does such kind of work. This neurological work studies. So I've done such work on those areas. But if you can have access to MOU, they can do metal ligand docking. Also, to explain better, there's a work I shared that came to my table this week on corrosion inhibition, where we have to use extract of a leaf, moringa, to inhibit maize tea. So we notice that the maize tea contains a good percentage of iron. So we want to use our ligands to see how it will prevent the corrosion. The corrosion, how we can inhibit the corrosion of the iron, which is the maize tea, because maize tea contains iron. Pyrus cannot do that because that will involve ligand metal docking. So you can use Material Studio. You can use Material Studio for it, which is still, you buy it because you get licensed Material Studio. I will even encourage every chemist in the house to go and look for Material Studio. Mostly those who are interested in material science. If you're interested in electrochemistry, if you're interested in electrochemistry, if you're interested in material chemistry, then you need Material Studio. You need Material Studio. So we can do that. So we are currently working on that work for that participant. Where we dock ligand with a metal. So all these are possible. It also depends on your accessibility to the right tools. Can you access the right tools? But they are possible. So if you have such work, bring it. We'll tell you what it will cost. Because it will be expensive now. Thank you, sir.
Okay,
Thank
sir.
you.
Where can we access the Material Studio, sir?
It's online now. It's online, but you pay for it.
Okay.
Go online, type Material Studio.
Okay.
I don't know the amount they're selling it now. Because why? The reason why I did not have that understanding is that, as a chemist, I'm not more in physical. It's those who
Material
are
science.
mostly in physical and inorganic. Then let me throw a little light. I don't know if you have done a work. I want to cancel something. There's something I want to delete now. I please just apology for what I'm about to say.
Please,
It might hurt
please
some
mute
of
your
you.
mic, please. I
It
cannot
might
hear
hurt some
what
of you.
you're
How many
saying.
of you have done a work on nanoparticle of plant extracts?
Excellent. Please mute your mic, please.
Has anyone done a work on nanoparticle from a plant on green synthesis?
Yeah, it's a work I want to do. Oh,
All right.
I
Now,
want to do
what
a
I'm
work
about to
on
say will hurt.
nanoparticles.
Yeah, what I'm about to say will
Okay.
hurt anyone in this category. When nanochemistry, when nanoparticle, when research on nanoparticle started, a lot of persons, when I see their work, I keep seeing green synthesis. Sorry to say this, all what you are doing is not scientifically proven. I call it rubbish. I will give you reason, and you see a reason with me. Last year I was at Boy University, a professor in Trinity University, Lagos, was there for a bioinformatics conference. This professor was working on nanoparticle using a plant extract. I told him, Prof, please cancel this work you are doing, except you don't want the work to go far. He said, why? I said, number one, this your work is not reproducible. It is not explainable. You can never explain it. If you use a plant extract and you want to form a nanoparticle, a metal complex, then you took maybe silver nitrate and complex with your plant extract. They call it green synthesis. Number one, how do you really know if, yes, you agree that it has formed a nanoparticle, but in the principle of nanoparticle, there must be a capping agent, a stabilizing agent, and also a reducing agent. So the question is, this extract you are using has about 100 and more bioactive compounds. Which of them in particular formed this nanocomplex? There is no characterization as a stands now that will tell you that. Because for you to form a nanocomplex, it means that a particular bioactive compound served as a ligand, and it might be a bidentate ligand or a polydentate ligand that got attached to the metal, depending on the coordination number of that metal. So do you want to tell me that all the bioactive compounds in your extract all acted as a ligand to form a complex with that metal? Which is no. So number one, you cannot explain the chemistry. Number two, you cannot give us a structure. You cannot propose the structure of that nanoparticle. And you know that anything in chemistry that cannot be presented structurally, like anything you produce, you form in the lab, an equation, you cannot write the equation of that reaction. You are doing nothing. So can you propose the structure of that nanocomposite, that nanostructure? You cannot, because you cannot tell me the bioactive compound that complex with that metal. And if you do your characterization, what do you want to do? This MS will still tell you the list of all those bioactive compounds there. ATR will still tell you the functional groups of the compound. And there are many compounds that will have the same functional groups. So you want to start doing TM, SEM, all those ones are for particle size characterization. But if you want to do structural characterization, there is no adequate tools that can tell you. So the best approach to do a nanoparticle is to use a pure compound. Okay, but even before I explain, you cannot still reproduce that work. Imagine you use a plant extract today and synthesize a nanoparticle. Use that same plant extract. You will not achieve the same nanoparticle you have synthesized. It must be different because of the diversity in the bioactive compounds, because of the diversity in the geography of that plant, the variations, the genetic variations where those plants were gotten. So if I develop a green synthesized nanoparticle using guava leaf today, and somebody wants to repeat that work in Kano, you will not have the same work. So if at the end of your research, your nanoparticle was very good, effective, what is your goal? Mass production now, reproducibility. You cannot reproduce the work. You cannot reproduce the work. So the best approach is to use a pure compound. Now, when you use a pure compound, you can now tell the denticity, the dentate nature of the bite of the ligand. You can now propose a structure of that nanoparticle. You can even tell us how many of the pure compound, which is a ligand, is binding to your metal, which all depends on the coordination number. You can now use docking. I don't know, but I have checked. I think to see people who, I don't know if you have seen any work on docking of nanoparticle. Now, we are privileged to be among some early scientists who applies docking with nanoparticle. Because before you do docking, remember that before you do docking now, you must have a structure. You must have a data, a structural file. So if you are using plant extracts, you cannot do that in docking because you don't have a structural file. But if you're using a pure compound, we can now compress that pure compound with your metal and propose a structure. Use that structure for docking. We also have a training on nano drugs, docking of metal complexes, and docking of nanoparticle. It's a different approach on its own. It's still part of our training for this month. Maybe I will just give you our calendar for the month, and you get yourself prepared and ready for whatever you see each month. So if you have a question to ask on this, you can. But with this viewpoint of mine, I want you all to change your mind from green synthesis. If you want to exploit scientifically, I rest my case.
Well, thank you very much. That was some good insight.
I was expecting reaction, no?
We are reacting. We are reacting.
Like a negative reaction.
No. Like I said, that was some good insight because last year I reviewed an article last year, and the person claimed that it's a she that she did a work on nanoparticle. You understand? So I told the person simply, there's no way you're going to work with a good extract of a plant and expect that you can do docking with it. How did you do it? So she claimed she did. That's okay. What you may do for me, send me the methodology and send me the references of work of this nature that have been done before using nanoparticle. And I can tell you, for weeks, she couldn't, send me any reference material on that. Eventually, she sent me one, and when I look at it, I noticed that what was done in that work was not even, they didn't dock the nanoparticle. Rather, they harvested the ligands from that of the extracts and from the GCMS analysis, and that was what they docked, not the nanoparticle itself that was formulated. So like you said, it is very difficult to carry out docking using nanoparticle that formulated from a mix of phytoconstituents in the plant is difficult. I've not seen any work like that. That was why I said something. That explanation was very, very nice. Thank you very much.
Thank you, Prof. Thank you. So, Professor Excellence, I'm sorry, please pardon me. Okay. What we'll do now, why? Because even if we continue, we will not definitely enter the pharmacokinetics. So, we are going to agree a date now, but we shall do we are going to visualize the docking stuff to see the 2D and 3D interaction before we go. So maybe I will drop the dates for the last class, since this class was not able to. We have a training starting 10th, and today is 6th. So let me see. Let's see on the 14th. 14th is Valentine's Day. Some of us should be somewhere. Or can we have it on Monday, 9th? Next week, Monday. How is our schedule?
It's okay.
So
It's okay.
It's okay,
I think
sir.
it's for Tuesday.
okay, because I'm not autocratic, I have to see your schedule, because our brain chemistry starts on 10th. While Wednesday 1st, Wednesday 11th is okay. We have 11th and 12th is free. So your choice, Monday or Thursday, whichever one you want to choose next week, Monday or Thursday.
Monday is preferable. I prefer Monday.
Okay, so we'll just do it on Monday.
Monday is fine.
By Monday, we'll come back. We shall focus strictly on the pharmacokinetics. I have a lot to explain on that pharmacokinetics. My problem is that I thought too much. That is all the problem, which I'm trying to reduce the way I talk. So I will try not to talk too much, but
Oh, so your talk it makes a lot of sense, so because it's enlightening. Your talk is enlightening, sir. So please don't stop talking, sir.
people like me does not cover stream of work. we don't cover the stream. I don't know why, but I have tried to cover stream in normal school teaching. Okay.
Too
What
much
we'll
knowledge,
do?
sir. Too much knowledge.
What we'll do now is we are going to do something with this result now. So first of all, we will sort these results. These results appear there. If you look the first one, 536, it appears up to 9 times. Up to 9 times from 0 to 8. From 0 to 8. That means this docking, as it was running, the protein was rigid, but the ligand was flexible. Yes, so the protein was rigid, but the ligand was flexible. So the ligand was checking, interacting at different positions in that active site to see the one that we have the lowest RMSD. So the one with the RMSD of 0. So what Pyres does is that Pyres estimates all lowest RMSD to 0. So the lower RMSD out of this 0 to 8 here. for the same compound, for the same ligand. So the lower RMSD, that is the result we shall pick. Now, but I wouldn't want to be stressing ourselves picking just the ones with 0, 0, 0. We are going to sort this result in Pires such that all the ligands that have 0 RMSD will be sorted. They will be at the top, which is the ones of interest, while every other one will come down. So for you to sort that, come to this binding affinity. Just click once on the binding affinity. Just click once. Once you click once on that binding affinity, go to mode, this mode. On that mode, also click once on the mode. Now, what did you notice? You would now see that the 7 ligands with a 0 MSD are on top from 1 to 7. 1, 2, 3, 4, 5, 6, 7. So our focus will just be on these 0 RMSDs. Now, we are going to save this result, but before we save the result, we are going to save the complexes. We'll save the complex, we'll save the, if you look here, you see 1086 hypertension with _129. So this is a complex between this protein and the ligand. So what you are seeing here is complexes. Complex means the protein and ligand being together. So now we are going to save some of the complex, not all. We'll save one, the native ligand complex, and we'll save our, for me, how I do save my own, because I told you that I picked the first 5. And these first 5, I will save each of them too. The first 5 ligand, the first 5 best, the first, how will I put it now? The best first 5 or the first 5 best ligand. I will also save their complex. But meanwhile, most people usually save only the best ligand, the ligand with the better inhibition. So the lower the binding affinity, the better the result. So this minus sign is telling you a lower binding energy, a very low binding affinity, which means that the ligand was able to, it has a very low energy requirement to bind to the protein. So the lower the value, the better the inhibition. And how will you know? Once you have a high minus value means better inhibition, because minus 9.6 is lower compared to minus 9.3. So the high negative value implies better inhibition, which is lower energy. That means it has a very low energy that it can easily interact with the amino acids in the protein. So what most people does is they will save the native ligand as complex and save one of the ligand of their test ligand that gave the better result compared to others. They will save that one as complex. But what I do is that I'll pick the first 5 and also save them as complex. So I'll be generating the 2D and 3D interactions for each of these first 5, because I don't put all my hope on that one with the highest binding affinity. But for the purpose of the training, I will just save the native ligand as complex, save one of our test ligand as complex. So you now know what we are doing. You can replicate it for others. There is no need saving the control as complex. They don't going to save the control as a complex because we are just using the control to compare the binding affinity, except you want to use the control to compare interaction. And that is why we are using the native ligand. We are using the native ligand to compare interaction. While we are using the control, the reference drug, to compare activity, to compare the binding energy. So it's depending on your concept now. If you want to also use your reference drug to compare interaction pose, and at the same time use it to compare activity value, you can still do that. So let's identify. So for those who were in the beginner's course, you will not notice that I'll be stressing myself to identify which compound is bearing this CID. But if you had converted the name, like what Dr. Abisoye sent to the platform, she was trying to edit to rename the download. And after renaming, the name was not still appearing. So that means we're using the other approach. So if you use the approach we discussed last week, you will now notice that the name will be appearing instead of the CID codes. That will ease up stress of identifying which is which. So let me identify our native ligand. I will just do this. Let me just shorten my screen. Then I will do this also. So this will enable me to just identify which one I'm. All right. So from here now, you can see that my native ligand is so from here you can see that my native ligand, what is the number? 3152. So this is my native ligand. This one, I will click on it. Just once you click on it, it will appear on the top here, this white space. So 3152 is my native ligand.
I thought it was 5361.
No, look at it. Donpezil. Oh, sorry. Donpezil was my negative ligand, not native. Okay, but we don't have business with it, yeah, so I can remove it. Sorry. Okay, 5362119. That is native ligand. 536. You can click on it, so it will appear up. 5362129 is my native ligand. So I will also look for my control. Remember, I use 3 controls, and I have already changed this man. Good. So I use 3 ligands, which are 131, 140, 129. And if you check now, you notice that 2 of my ligands are at the top. So you see why they're having a better result? Because we screen them based on drug-like according to the macrophoric features that are necessary for interaction. So it simply means that if you are working with drug-like molecules, you should be expecting better results. So one of the heat here, 9.6, is from my test ligand 129125. So I will just, if I were you, I can save 3 of them. The first one, 129, the other one. So you can also save the other one and the first 5. So that is what you can do. Now, if you notice, our drugs, our reference drugs, we are below 5388. And wait. I'm making a mistake. 5362119. 5362119.
That's the native
Okay.
ligand.
Yeah, he's there.
Great.
He's there. So let's save this now. So this is 5362119, which is our native ligand. You right click on it. Once you right click, want to save it as complex, click save as PDB. Once you click save as PDB, you go to where you want to save it, maybe your download, then you type just say native ligand complex with 1086 hypertension. So you just say native ligand complex with 1086 hypertension. So this will tell me that that is for my native ligand. That's a complex. You click save. Then the other complex you will save now is your test ligands, the one that they for me, I will save the first 5. But for the sake of the training, we just save one, which is the highest. Highest here is 129. And this 129 is from my structure, for my test ligand. So I click on it, it will appear up. 129, that is this last one. 129.25621. Oh, what happened there? Okay, so 129.25621. So once you have clicked on it, it will appear up. Then go to that top, right click and say save as PDB. Choose where I want to save it. The same thing, can change the name to the code 129.125.621 complex with hypertension. You save. So we have saved the complex we want to use. So if you want to save others too, you can save the 131, click on the 131, it will appear. You also right click, save as PDB 131 complex with So now I will save this Excel result. This my table here will be saved in Excel. I will save it as an Excel file. Just after where you clicked on insert item, come to the 4th icon. The 4th icon is saying save as comma separated file, that is CSV. Save as CSV. So we are saving this result, this sheet. So we click on save as CSV, go to where you want to save it, and you type whatever I want to use to save it. I will prefer molecular docking results of what? The plant. My plant is peperomia. Peperomia pellucida. On there, sorry, 1086 or with 1086. Whatever I want to save it, that is also your business. So saving now is left for your choice. So you click save. So we have done all the necessary savings we want to save. You can open the results. When you come back to your table, to your download now, you can see the document results. So you can open in Excel. You can open in Excel. So this is the docking result. This is where we are after. This is what we are after. You copy this, you can come somewhere fresh. So this is what we what we are after. So we don't need this IMSD again because they already zero IMSD. So we don't need them again. Then you have to put your units, the binding affinity, kilocal per mole. What I usually do is that I do turn this ligand to PubChem CID. Then I do have my ligand now beside. So what you just have to do is you have to on your own. So remember, when you if you did multiple, maybe you splitted your compounds, then you join all of them that have 0 RMSD from different files. Like you can copy again from another file, come below and paste. So you join them together to become a single file. Only the ones with 0 RMSD. Then you have to remove these hypertension stuffs here by just click. You remove like this because the results you present to us will not be all these UFF and hypertension. So you do this for all. Okay, you do this for all the compounds you docked. Then get the name of these compounds now. Take the CID to PubChem, copy the name from PubChem and paste under ligand. Because under ligand, what you have here will be the names of those compounds. Why here will not be their CID? Are we together? Hello?
Yes, sir. Yeah, together, sir.
All right. So what I said is you do what I'm doing. I'm just deleting all this. Because what will be as your doctoral results, you have seen from paper, so you know what I'm saying already. So you now you would go to PubChem now, copy this CID to PubChem, get the name, the common name, or the IUPAC name. The IUPAC name might be so long, get the common name, paste under these names. So this is your docking result, and your result table. Then you can save it here, or you better do this on a different sheet. So you can open a different sheet to keep it separate, so that we know that is different. Okay. Now the next thing you will do now is to visualize your complex. So these are the complex 129 complex with and the native ligand complex. And please, if you are doing this, well, you can go to literatures to see how docking results are presented. Because from this name now, you're going to indicate in brackets the ones that are control, negative control, negative ligand in brackets after the name. It all depends which way you want to represent the result, but literatures now will help you. Then you open your Discovery Studio now, because we shall use it for the visualization. Now there are 3 things I will do here. I will do superimposition. If I see a paper on docking without superimposition. I do throw away that paper because it means there was no validation, like in slippery validation. The essence of that native ligand is to validate the binding interaction of our own ligands. So we are expecting that where the native ligand binds should be where our own ligand should bind to achieve better results. So we are going to superimpose the native ligand with our own ligand. So here, I only saved one of our ligand as complex. So assuming I saved 5, that means I will do each of them, 5 of them, one by one with our native ligand. So let me quickly show you. Let me show you how to do your superimposition first to know if where the native ligand binds is also the same active site with my own compound. Are we together? Hello, are we together?
Very well.
Yes, sir. Yes,
Yes,
sir.
but yeah.
Okay. So quickly go to file, click on file. Once you click on file, click on open. When you click on open, locate your protein, the prepared protein. The prepared protein, open it. So once that is done, this is our prepared protein. Which one is this? All right, so this is our prepared protein, the 1083 hypertension. Change the background to white. By go to color, right click, go to color, go to background, and click on white. You say okay, and it will turn white. Then come to the left, you see this drop arrow, this little arrow. Click on it to open up the panel of that protein. So this is our wonderful protein. Now what you will do now is this. Bring in your complex. You don't bring all at the same time. You bring in the complex of our native ligand and one of our heats. So if I have so many 5 heats, I'll do them, each of them against my native ligand one by one. So go to file again. Click on this panel, this protein panel, the panel showing the details of this protein, which is here. Click on it. Go to file. Once you go to file, you see insert form. Insert form. This insert form. Click on file. Then highlight the ligands, those complex, the native ligand complex and my heat complex. Both of them, you can select them together. Then you open, click on open. You will see both ligands we show here. One is checked. The one that is checked is our heat ligand, and one is unchecked. So if you want to rotate this protein, hold your right mouse. Once you hold the right mouse, you can now turn the protein. So as you are rotating the protein, you will see the structure of that your ligand. If you want to change the mouse functions, you right click. When you right click, you go to mouse mode. You see rotate, translate, zoom, torsion. So zoom is to increase the size. Translate is to move it from one point to another. Rotate is to turn it around. But you can rotate by just holding your right mouse and be turning the structure. So what we'll do next is click on the native ligand, check it. Also click on it. So once it shows good, let's see if both structure we are at the same position. So I can now zoom it. Let me pick my zoom. Zoom. So this will tell if my heat and the native ligand, if they were at the same position. So what can you tell from here? What can you tell? So this is my native ligand. Why this is my heat ligand? So who can explain something from here? And I just little.
They appear to be in the same coordinate.
Good.
And
That's
this
just
side
what I
by
need.
side.
Yeah. So assuming they were far from each other, that means they were nowhere superimposed. It simply means that my own my heat did not even bind at the same binding site with the native ligand. So this is a better result. Very, very good result. But I'm going to give them different color so that I can know which one is my native ligand and which one is my heat, which one is my native and my heat ligand. So assuming I have 5 heat ligands, I will do this for each of them. So what you now do is to give it color, because I have to change the colors. So if I see a docking publication without this superimposition image, I throw it away, because you did not tell me if your heat actually, if they were docked at the same site with your native ligand. So we know, we call it docking validation, to validate your docking. So you validate your docking using native ligand. So what you do is come to this first one, which is my heat ligand, right click. When you right click, no, let me see, click on it again. Good. Right click. When you right click, you will see attributes. Click on attributes. Now, from the attributes, you can even see the SYZ binding interaction. 43.77, 32 point this, 42 point this. So you can compare this coordinate with that of the native ligand when we do for native ligand, if they are close to each other. So even without looking at the structure, you can compare the SYZ coordination, see the slight variation. So I want to change the color. That's my point. Go to color. Double click on color. Once you double click, another color icon will appear beside at the right. Then you can see 3 dots. Click on that 3 dots. So choose the color you want to give it. But mind you, look at the background already. The background is having red. So let me say blue. Blue for my heat ligand. I would choose blue. Then I will say OK. Then click apply. Then click OK. So you now see that my own heat ligand is showing blue. So under the title, the image title, I will indicate blue for heat ligand for so name or heat ligand. Then do the same thing for your native ligand. Right click, go to attributes. I'm really sorry I'm taking much of your time, but I think in the next 9 minutes we should be over. Okay, do you notice something here? We're able to recall the SYZ of the heat ligand.
Yes, they are very close.
Good. So
That one was
even without
4.3777, and this is 368 and
all
0.69.
right,
And then
now
that one
that is
77.
just to tell you more that this whole interaction, we are the same binding site, and it's a good work. I wish I can even do something from here and publish it. Then click on this color, click on these 3 dots. Let me say I'll give it yellow. I'll give it yellow. I'll say OK, apply, then OK. All right. So you see, there's a difference now, and you can now look at. In fact, it's as if they are even intertwined, as if they are trying to like they are twisted. So I will change this graphic image. Go to script. Script is a menu bar. At the menu bar, click on script and select visualization. Select visualization. Click on publication quality. Publication quality, and it will brighten up the image to give us a better image. Do you love what we are seeing now? So this is quite nice and better. Save this image. Save this image. I can equally change it now because since I'm seeing blue ribbons, I can decide not to use that blue again. Since I'm seeing blue ribbon now, after I adjusted it, I can use red.
This my color is not reflecting on. Trying to change these colors. Not reflecting in my stuff.
Mm. It's not reflecting.
Not reflecting on.
See what you're doing now? You right click. When you click, you right click attribute,
Yes.
on color. Click on this first one. Once you click here, to bring up this one behind. Can you see my screen?
I can see your screen.
Then click on this one. Choose the color you want. Let me choose purple, this color. If you choose this color, you click OK. You click OK. Click apply first. After OK, click apply. Then click OK again. The color will change. Look at mine too. Mine have also changed. So
No, it's not changing. No.
maybe you can try it sometime. It's not difficult to change. Let me keep it back at red. So now my red color is my heat light down, while my yellow color is my native light down. So you can keep the color consistent. That means that is if you use the other heat light downs. So let's save this. Once you save this, you go to file. Click on file. When you click on file, click on save as. Click on save as. Now, if you click on save as, change this Discovery Studio, this save as type. Save as type. Change it from Discovery Studio. Choose image files. Choose image files. Image files. Once you choose image files, then give it a name. Name it. I will call superimposition image. So you're learning all this just for 20,000. Superimposition image of yes, native light down and test light down, native and heat light down. You can specify the heat light down with 1086 hypertension. You click on save to bring up another panel, click OK, and that image has been saved. You can go to your download. You can see the image, superimposition image. You can open it. So the image will be part of your docking work. So having done that, we are going to now do the 2D and 3D visualization. So this is a beautiful image. That's by the way. So let's now go back to our Pirates. So to our service studio. What I will do now is to I'm going to remove to avoid, let me see this will work. To avoid the issues, delete everything. So delete. Okay, no, just close it and reopen it again. Just close this one, because most times you may encounter problem. So go back again, go to file, open your protein. So that will not take you time. Open the protein, remove the color like we have done. Remove color you have done. Open the protein panel. Click on the panel. Then go and get your. Now, I will even advise you to also generate 2D of your native ligand. 2D and 3D of your native ligand. 2D and 3D of your heat ligand. So that you can also check if the amino acids residues that we are involved in the interaction of your native ligand. Since we are saying that our native ligand and our heat ligand, they had similar coordination, similar coordinates. So let's see if there were some similar amino acid residue that also took part in that interaction. So these are what you explain in your doctoring results. So it starts from, I'll pick, at this point, you can't do both of them at the same time. So let me pick my heat ligand first. My heat ligand, I open. So when that is done, come here, come to show 2D diagram. This show 2D diagram, click on show 2D diagram. So you can imagine that this is the compound, this empty, dry compound that was responsible for that high binding result of minus 9.6. If you are told that this compound will have a better binding result, nobody will agree, seriously. Even me, I will not agree. Because you're expecting to see, I want to see some things now. But thank God it has the binding interactions are actually weak, but it has a conventional hydrogen bond as a result of the amino group here. So that is still okay. In our Monday class, we shall explain these interactions. We shall explain these interactions a bit and the implications. So save this your 2D. Save this 2D. You go to save as. You say save as. Now, don't bother yourself. It's already at image files. Assume it was at Discovery Studio, then you change it to image files. Then what's the name? What name do you want to give to the file? I can call it one. So that is 12912 whatever 2D interaction. I'll call it 2D interaction or 2D. You just left on you. 2D interaction with. So you decide what you want to save it. Then click on save and click OK. Then that is done. Come to the 3D now. Click on this protein at the middle. The structure, the protein structure at the middle. Are we together? Ewo, I think I should just stand up here. I will end here, sorry. This is 10:31, so I'm going to end here. So this at the middle, then come back to this panel, choose ligand interaction. Click on ligand interaction. But then you can close this your 2D. You can close that one. So once you generate the 3D here, at this point, click on, you see a hydrogen bond. this hydrogen bond below display receptor surface at the right, hydrogen bond. Click on the hydrogen bond to show the hydrogen bond cloud, the hydrogen bond cavity. So what you will do is right click, you see labels. You want to show the amino acid residues. Click on labels. Under object, change it from atom to amino acid. You can change your font. I prefer using banana.
The labels, you got it from where? I can't see the line in the labels. Labels about.
You say?
You're talking about labels just now.
I didn't get you.
Labels you just mentioned.
I'm not hearing you clear.
Labels you just talked about now.
Okay, yes. The labels are the amino acid residues to show them. Just right click anywhere. Right click. You see labels down. On that label, click on add labels. Then you see object. Below the object, there is atom. Change this atom to amino acid. The first amino acid, not one will change. This amino acid. Click on the amino acid. Leave this one as name. Leave it as name. You can change your font from Arial to prefer banana, so it will be a bit bold. You can change the font size. Prefer 14. Then you click apply. It will show. Then click OK. Now, if you watch this 3D image is too large, you can minimize it by change your mouse mode to zoom. Then minimize it. You can rotate it to show the cavity. You can just rotate it to your choice to show the structure inside the hydrogen bond surface. Like this is better. Then go to script as we did before. Go to script. Then do your visualization. Click publication quality. So to change the image to give us good quality for publication. So I prefer this. Sorry. You can now save. Go to file. Go to save as. It's showing Discovery Studio files. Change it to image files. Then copy this and rename it to 3D. So the 3D. You save. So at this point, you are done with your post-docking analysis. Now you can equally want to generate the same thing for the native ligand, no problem. So that so you can compare the amino acid residues if they could be similar. So we have successfully did what we are supposed to do. So what I owe you now is pharmacokinetics. So these are our image. These are our image. I really want to appreciate you for pulling through to this time. Eight good sessions. 8 good sessions, and you are always available. You can copy this image to your documents. Then we shall look at the by Monday, as we have agreed, first 1 hour to interpret these results, this 2D interaction. Because if I'm going for conference, if I'm presenting my paper in a conference, I usually spend time to discuss what this whole interaction means. Then we should also look at picking, and that will be the end of this. So I'm giving you extra 1 day. You should pay me for that one, but I'm just doing it. We are 18 left. Here we It's not easy, Sha. It's not easy. Am I expecting question? If you have, you can do it to ask.
Dr. Henry, are you
Yes,
there?
sir. Yes, sir.
Well, mine is not a question, but it's to appreciate you for taking this time. I think the training is very thorough, by my own judgment, and I want to appreciate you. Thank you very much for taking this much time to go into this more details with us. And thank you very much. So we appreciate if you can put up that 1 or 2 hours for us on Monday, as you promised, so that we can completely or fully grasp the concept. And also, we expect that all the videos will be released so that maybe in our spare time we can also go through those videos. Thank you so very much. We appreciate it.
Thank you. Thank you so much, Prof. Thank you. I really appreciate everyone. It's not easy, but currently since this week, if you notice carefully, I have it's as if I'm struggling with my breath a bit
Yeah,
because
got a
not
cold,
been that
kata
too
something.
not been that too strong, but we still want to push through. So we are pushing through, taking medications and having some rest.
Yeah, it's
Yeah.
needed. Thank you very much. Did you take some of these herbal preparations? Yeah,
I think
maybe
I will
maybe
take some of
some.
our herbal concussions.
Yes, right. Maybe your guava leaf and scent leaf and
Yes, yes.
anona moricata, all those combinations and prepare concussion.
There
uh,
is there is something my wife said. She said, I don't take drugs, but I produce drugs. How come? I don't like I don't like taking drugs. But every day I preach about drugs. Hey,
all
that
right.
is
Thank you. I
that is where
am
we are find ourselves.
I'm very tired. I think I
Yeah,
want to
we
take
want to
a rest
appreciate
now.
everyone. It's
Thank
not
you.
easy. It's not
Thank you
easy.
very much,
So
sir.
yeah, we'll say good night.
Good night. Thank you.
Say good night.
Yeah.
So thank you. See you all Monday. So continue doing your work. Continue your work, your personal work. Just continue. Continue. Professor Justina Otombon, we are welcome. Thank you for being available. All right. We are 10 left. It's not easy. Thank you. To see you all Monday. So if you have questions, just write it down. That Monday we may have time to interact a bit. On Wednesday, we're having a webinar. We'll look at how that would be. Good night.